Hi, I'm Monisha and I'm going to speak about the instrumental analysis of food. So first, what is food analysis and what is the need for it? So food analysis is a discipline dealing with the development, application and study of analytical procedures for characterizing the properties of food and its constituents. So for all food products, whether it's raw or processed, it is important to analyze and provide information about the different characteristics, including its composition, structure, physiochemical properties and sensory attributes. And in fact, the food is analyzed for many of reasons. For example, compliance with legal and labeling requirements, assessment of product quality, determination of its nutrient value, detection of adulteration, for research and development. And food safety is a major issue. And with growing concerns about the health safety, many of the food regulatory authorities in different countries have imposed many mandatory norms to find out the presence of toxicants, which if present beyond a prescribed limit, it can be hazardous to human health. So many of the government bodies regulate the permitted levels of contaminated compounds. To find such level of the compounds, we use some analytical instruments. So next, what are all the criteria used for selecting a good analytical technique? First is precision. The technique used should be precise and it should give a good result by specific analyst using the same equipment and experimental approach, keeping other conditions unchanged. Next is reproducibility, a measure of the ability to produce good result using the same experimental approach in the same as well as different laboratories using same or different equipment. Next is accuracy, a measure of how close one can actually measure the value to the true value of the parameter being measured. Next is simplicity of operation, a measure of the ease with which relatively unskilled workers may carry out the analysis. Next, the equipment should have a good speed. Even in a given time, it should analyze single sample or number of samples in a given time. Next, sensitivity, a measure of the lowest concentration of the component that can be detected in a given procedure. Next, it should have specificity, a measure or ability to detect or quantify specific compounds within a food material even in the presence of similar compounds. For example, it should detect fructose in the presence of other sugars like sucrose or glucose. Some of the important instrumental techniques used in food analysis are gas chromatography with mass spectrometry, high performance liquid chromatography, UV visible spectroscopy, Fourier transform, infrared spectroscopy, and differential scanning calorimetry. So first we are going to discuss about the gas chromatography. First of all, chromatography is defined as a process by which solutes are separated by a dynamic differential migration in a system consisting of two or more phases, that is mobile phase and stationary phase, in which the particles are separated based on their differences in absorption, partition, solubility, vapor pressure, molecular size or ionic charge density. Then gas chromatography is one of the most versatile analytical techniques used in the food industry and which is used to separate volatile organic compounds in a mixture, and it enables fast separation and identification of the components in a complex mixture using appropriate detectors. And in the schematic diagram we can see the various components of the gas chromatography, where the samples are rapidly injected by means of the micro syringe through a silicone rubber into the column. Then the sample injection port, column and detector are heated to temperatures at which the sample has a vapor pressure usually about 50 degrees or above the boiling point of the highest boiling solute. The injection port and detector are usually kept at higher temperature than the column to promote rapid vaporization of the injected sample and prevent sample condensation in the detector. The separation occurs as the vapor constituents equilibrate and partition between carrier gas and the stationary phase. The carrier gas is a chemically inert gas available in pure form such as argon, helium or nitrogen, and the response is usually detected in form of peaks. The sample is automatically detected as it emerges from the column at a constant flow or at gas pressure rate, using a variety of detectors whose response is dependent upon the composition of the vapors. The chromatographic peaks are recorded as a function of time. By measuring the retention time, the time a sample is injected and the time between the chromatographic peak reaches the maximum intensity, and comparing this time with that of the standard of the pure substance, it may be possible to identify the peak and to identify a compound. These are some of the applications of gas chromatography. First, cholesterol, fatty acid profiling and transfer analysis for the detection of antioxidants and preservatives like TBHQ, benzoic acid, sorbic acid, acetic acid, etc. And for the detection of residual pesticides and environmental contaminants, for the characterization of flavors and fragrances, and the gas chromatographic profiling of the essential volatile oils gives a reasonable fingerprint which can be used to characterize the identity of the particular oil. Next is one of the widely used technique called gas chromatography with mass spectrometry. The GC portion of the system provides separation of the volatile organic solutes in a mixture in a gaseous phase. As each solute exits the GC column, it is diverted into a mass spectrometer, which is capable of monitoring both the amount and identifying the chemical nature of the solute. In this way, both quantitative and qualitative information about the mixture can be obtained. Next, the MS portion of the system takes each solute eluting out of the gas chromatograph and ionizes it in an electronic beam. The ions formed in a specific solute will depend upon the nature of the bonds of the molecule, and it is possible to get both ionized molecules and its fragments. The ions thus formed are then directed down a separator which isolates and counts the ions according to the mass. The sequence and relative intensity of the mass peaks gives information about the chemical identity of the solute. The absolute intensity of the peaks provide information about the amount of substance present in the food. These are some of the applications of it. First, to analyze the pesticide residue in all processed and food products, to analyze the environmental contaminants in food products such as polychlorinated biphenyls, polyaromatic hydrocarbons, dioxins, etc., fatty acid profiling in oils and fats, to identify the flavors and fragrance in food products, and to detect the volatiles and other residual solvents in food packaging material. Next analytical technique used in food analysis is high performance liquid chromatography, in short HPLC. HPLC is the advanced version of liquid chromatography. HPLC is used to separate, identify and quantify polar and non-volatile compounds when in mixture. HPLC utilizes a column that holds liquid stationary phase, a pump that moves the mobile phase through the column, and a detector that shows the retention time of the molecules. HPLC is an excellent technique for separation of chemical compounds with high degree of specificity and selectivity, and is basically a highly improved form of column chromatography. Compounds are separated by injecting a sample mixture into the column, and the different compounds in the mixture pass through the column at different rates due to differences in their partitioning behavior between the mobile and the stationary phase. The most commonly used stationary phase is silica. As a sample solution flows through the column with the mobile phase, the components present in the injected sample solution migrate according to the non-covalent interactions of the compounds within the column. In order to detect the components as they elute from the column, there are many types of detectors that can be used with HPLC. Some of the common detectors include ultraviolet, photodiode array, refractive index, and mass spectrometric detector. And some of the characteristic features of HPLC are fast, accurate, and high power of resolution. Results are repeatable and reproducible. They facilitate determination of multiple components in a single run. They provide method of choice for thermally unstable and high molecular weight compounds. And the separated compounds can be easily collected from the mobile phase for further characterization, and it is a non-destructive technique. HPLC finds wide applications in food, both for profiling and analysis of various components such as amino acid profiling, peptides and proteins, lipid and alcohols, carbohydrates and carbohydrate profiling, sweeteners, fat-soluble and water-soluble vitamins, carotenoids, organic acids and organic bases, residues of mycotoxin, antimicrobial and veterinary drugs, pesticides, etc., pigments, colorants and phenolic compounds, bittering substances, and for preservatives, antioxidants, and stabilizers in processed food products. Next is one of the commonly used analytical technique, that is UV visible spectroscopy. It refers to the absorption spectroscopy or reflectance spectroscopy in the ultraviolet visible spectral region. It is an analytical method that can measure the analyte quantity depending on the amount of light received by an analyte. A beam of light from a visible or UV light source is separated into its component wavelength by a prism or diffraction grating. Each monochromatic beam in turn is split into two equal intensity beams by a half mirror device. One beam The sample beam passes through a small transparent container, that is cuvette, containing a solution of the compound being studied in a transparent solvent. The other beam, the reference, passes through an identical cuvette containing only the solvent. The intensities of these light beams are then measured by the electronic detectors and compared. The most commonly used solvents are water, ethanol, hexane and cyclohexane. Solvents having double or triple bonds or heavy atoms are generally avoided. UV visible spectroscopy is the cost-effective, simple, versatile, non-destructive analytical technique suitable for a large spectrum of organic compounds and some inorganic species. Next is Fourier transform infrared spectroscopy, in short FTR. It is an analytical technique used to identify organic, polymeric and in some cases inorganic materials. The FTR analysis method uses infrared light to scan test samples and observe chemical properties. The FTR instrument sends infrared radiation through the sample with some radiation observed and some passed through. The observed radiation is converted into rotational or vibrational energy by the sample molecules. The resulting signal at the detector present a molecular fingerprint of the sample. Each molecule or chemical structure will produce a unique spectral fingerprint, making FTR analysis a great tool for chemical identification. And FTR spectroscopy is an established technique for quality control when evaluating industrially manufactured material and can often serve as the first step in the material analysis process. A change in the characteristic pattern of the absorption bands clearly indicates a change in the composition of the material or the presence of contamination. If problems with the product are identified by visual inspection, the origin is typically determined by FTR microanalysis. This technique is useful for analyzing the chemical composition of the smaller particles, typically 10 to 50 microns, as well as larger areas on the surface. FTR analysis is used to identify and characterize unknown materials such as films, solids, powders or liquids, and to identify contamination on or in a material, for example particles, fibers, powders or liquids, and to identify additives after extraction from a polymer matrix, or to identify oxidation, decomposition or uncured monomers in a failure analysis investigations. So this is about the instrumental analysis of food. Thank you.